it may surprise you, but actually about 3 million people around the world have a rare bleeding or platelet disorder. That's quite a few people. Now, you've just heard about hemophilia. That's one of the most well-known examples. But there are actually dozens of different disorders that involve abnormal bleeding. And there are many, many yet to be discovered. And you may wonder, well, is this only relevant for patients with these rare disorders, each one being individually rare but collectively affecting many people? And the answer is not necessarily, because platelets are actually involved in some way or another in a huge proportion of deaths in Western society. So if you look at heart attack and stroke, platelets are involved over there. So the more we can learn about how platelets function and the role in rare diseases of platelets, then perhaps we can learn something about these more common conditions. So the NIHR Bioresource for Rare Diseases in the UK, with many collaborators around the world, have engaged in a pretty large genome sequencing study. And the process is basically to collect samples and enroll patients from around the world with particularly rare disorders. One of them is the bleeding and platelet disorders subgroup, but there are other patients with immune deficiencies, pulmonary arterial hypertension, etc., etc. And the reason for putting together all of these different patients is that we can get uniform genome sequencing. So there's no batch effect. We can really get very nice data and at a very good price as well. So once we've collected the samples and we've done the genome sequencing, we also need to record all the detailed clinical features of these patients. So, symptoms, laboratory test results, whether there's a family history, because we're looking at heritable disorders. And so collecting the data and then recording it systematically in a database is one of the big challenges as well. And we've worked on particular types of coding systems to record these symptoms in a very systematic way. And the analysis, which is the bit that I'm involved in, involves looking at whether there are commonalities, so clinical commonalities between little subgroups of patients who also have commonalities in terms of their genetics. And when we do that, then hopefully we can discover novel causes of disease and potentially further down the line look at some treatments, of course. So far, this is an ongoing project, but we have already published a few papers on some new findings. So we've discovered a new genetic link between very large platelets, which are too few in number, and can result in some bleeding as well. And it goes hand in hand with deafness. So there is one gene which is quite well known, which is called MYH9, and that is the classic large platelet, few in number, with hearing loss, also kidney problems and so on. So this is really the second main gene in which you've got hearing loss and macrothrombocytopenia, which is this platelet disorder, going hand in hand in these families. There are currently 3 families which we know of, and the mechanism has been worked out to some detail. Another example that I can give you is a genetic change in a very well-known cancer gene called SRC. So SRC is actually the first ever discovered oncogene. You can find publications dating back to 1911. The first examples involved an avian flu virus, which contained this gene and then induced sarcoma in chickens. So it really dates more than 100 years. But until now, no germline, so no heritable variants had been found in this gene. And what we found was a gain of function variant in this gene, so it becomes hyperactive. And if you look at the symptoms in this family, what you have is very fragile bones with, for example, teeth fractures. You've got fibrosis of the bone marrow, low platelet counts, and some bleeding as well. A third example involves the gene called TRPM7, and this is a new genetic link between, again, large platelets and also heart rhythm problems. So, and in this particular case, because the mechanism has been worked out to some detail, it appears as if magnesium supplementation could potentially be some kind of treatment looking into the future. But importantly, apart from just finding novel causes of disease, we've also been able to extend the repertoire of variants in already existing genes that were already known about. But in some cases, there were only maybe 1 or 2 reports in the literature, and we've been able to expand that in some cases by 2 or 3-fold. So, for example, the actinin-1 gene, which causes macrothrombocytopenia again, so large platelets, few in number, CYCS, cytochrome C, that's another example, and many other genes. So this is also a sort of second benefit of this study. So just to conclude, I think that bringing together the genetic and the clinical data from hundreds of patients worldwide, and I stress worldwide, you need worldwide collaboration because rare disease patients are, of course, rare, and therefore you need to collaborate across the world to get the sufficient numbers to discover the cause of disease. So that's essential to discover new variants. It is important also to improve our basic understanding of platelet biology and perhaps bring benefits to the millions of patients with heart attacks and stroke. And another important point is that within months of publication, we can take these genes and incorporate them into a diagnostic platform. So this in the past would have taken years, but some of the genes that we've already published are already in our diagnostic platform. This is called the thrombogenomics platform. It covers all of the genes which are currently known to be involved in bleeding and platelet disorders. It's available to anyone in the UK, but actually also worldwide this year. We would really welcome anybody who has got a sample from a patient with a bleeding or a platelet disorder to send it across to us, and we'll be happy to do the targeted sequencing in the known genes. Thanks very much.